Good morning and welcome to the 2019 Fankuchen Award presentation and lecture. So Pat and I would like to share with you our heartfelt thoughts about this year's awardee. First, a bit about the background of the award. The Fankuchen Award was established in 1971 to honor the memory of Isidor Fankuchen. Fan, as he was known, was professor of physics at the Polytechnic Institute of Brooklyn from 1942 to 1964. Upon Fan's death in 1965, Jose Dane wrote an eloquent memorial. Quoting parts, among his scientific contributions are many crystal structures. He was especially concerned with macromolecular compounds and with polymers, with low temperature studies and with instrumentation. From 1948 on, Fankuchen served as American editor of Acta Crystallographica. He created the Polycrystal Book Service. Fankuchen was a member of two scientific societies, the American Society for X-ray and Electron Diffraction and the Crystallographic Society of America. These two societies merged into what is now the ACA, and Fankuchen served as ACA's first president. One particular part of Dane's memorial illustrates what it must have been like to study under Fankuchen. First and foremost, Fankuchen was a teacher. He taught all the time. But what a magnificent teacher he was, what forceful drive, infinite patience, and contagious enthusiasm. The ACA Fankuchen Award recognizes contributions to crystallographic research by one who is known to be an effective teacher of crystallography. Starting in 1971, when the award was first presented, and every 3 years hence, the Fankuchen awardee joins a growing list of distinguished crystallographers who also are outstanding teachers and mentors. We are delighted to announce that the recipient of the 2019 Fankuchen Award is Ed Lattman. Ed embodies the spirit of the board, both in his scientific contributions to crystallography and in his teaching and mentoring. As a mentor at Johns Hopkins School of Medicine, Ed was our thesis advisor. He nurtured our development as he taught us with great clarity the fundamentals of crystallography. He challenged us to seek innovative approaches to complex problems as we sought to shed light on protein stability and folding. He strongly encouraged collaboration. Indeed, pretty much everyone in the lab collaborated with someone in the basic sciences at Hopkins Medicine. After we graduated and left Hopkins, Pat and I ventured on our own, building our careers with the integrity, thirst for discovery, and drive for accuracy and precision that Ed inspired. About 20 years later, when Ed was leading Hauptmann Woodward, and I was leading MCCAT, I had the fortunate opportunity to work with Ed again when the MCCAT contract was awarded to Hauptmann Woodward. This time was different, though. We were colleagues, yet Ed continued to mentor, focusing in the areas of strategic planning, management, diplomacy, and as well as exploring frontiers and developing new technologies. Ed, through his leadership and passion for advancing knowledge across a broad spectrum, challenges us at MCCAT and within the MCCAT companies to embrace lofty goals and to expand our thinking. Ed is eminently deserving of this award.
Thanks, Lisa. So I was Ed Lattman's first graduate student, and a lesser man than Ed might never have recovered from that. But in fact, Ed recovered in grand style, and I'd like to tell you a little bit about his career. So Ed began his crystallographic training at Johns Hopkins in the laboratory of Warner Love, who's shown here. And so he's one of a number of extraordinarily talented crystallographers to emerge from this laboratory. Here's a picture of Ed from around that time, and I've included it because it shows him in conversation with another Fankuchen awardee, Lyle Jensen. So during his thesis time, Ed made a number of fundamental contributions to the nascent field of protein crystallography. For example, together with Wayne Hendrickson, he devised a concise way of expressing phase probability distributions. that is still in use today, and which everyone, aside from Wayne and Ed, refers to as the Hendrickson-Lattman coefficients. Ed was the first author on what I believe was the first description of what we would now recognize as the modern approach to molecular replacement. And given that we live in a time where probably 3/4 of all new protein structures are determined by molecular replacement, it's important to remember that Ed was in the vanguard. Ed also was able to introduce a methodological improvement in which he came up with an innovative way to parameterize the orientation angles that we use in the rotation function. So prior to his work, people relied on the angles that had been laid out by Leonard Euler, but under certain circumstances, those become correlated with each other and they lead to pretty nasty, messy solutions. And so Ed's approach to this fixes that. Now I want you to stop for a minute and think, how many scientists, let alone how many graduate students, ever find themselves in a position where they can say, in your face, Euler, it's rare. So after some postdoctoral stints that I'll skip over because I don't have time, Ed landed back in Baltimore, this time at the School of Medicine at Johns Hopkins, and this is where Lisa and I first encountered him. So during his time in the med school, Ed contributed to a number of important problems, and his work ranged from protein crystallography to small angle scattering to protein structure, protein folding, protein stability. He was also the editor of the journal Proteins, which is a post that he took over from the founder of this journal, Cy Leventhal. At some point, he moved back to the Homewood campus of Johns Hopkins, where he served as the head of the Department of Biophysics, and then later in university administration. Craving new challenges, he then moved to Buffalo, where he became the CEO of the Houghton Woodward Institute, and as we've heard from Lisa, this is where he began his relationship with IMCACAT. Most recently, he was the PI on a successful NSF proposal that supports much of the cool science on bio-XFEL work that you're hearing at this meeting. Somewhere along the way, he managed to find time to produce 2 very highly regarded textbooks. And I had the great good fortune to collaborate with Ed on one of these books, which means that, like Lisa, I had the privilege of knowing Ed both as a mentor and as a colleague. So this is all very impressive, but the Fankuchen Award is about more than this. It's also about teaching, right? And so when Lisa shared that photo with me of Fankuchen showing his students how to use a procession camera, it immediately reminded me of how Ed taught me to use a procession camera, one only slightly newer than the one in this picture, I think. Ed was a very generous and patient mentor. He gave you enough space to make mistakes, but he didn't let you drown. And I remember being able to go into his office, and it was like drinking from the fountain of knowledge, right? He had this uncanny ability to just take out a blank sheet of paper and a pen or a pencil, and he would derive from first principles pretty much anything you needed to know. So he was great one-on-one, he was great in the classroom, but his teaching goes beyond that, right? He also was always outward looking to the community at large, trying to share insights with the broader community. And so I think his textbooks are just one example of this. For this reason, I think it's appropriate to finish this bit up, not just with my words or with Lisa's words, but with words from other people from the community who have supported Ed's nomination for this award. So for example, we have a quote here from someone who had the opportunity to hear Ed's presentations, who says that you always learn the most when Ed was at the blackboard. Right? Other people have mentioned how generous and exuberant Ed is when he shares his insights with the community. Right? And because of his continually sharing these insights, he has made, I think, a significant contribution to the thinking of all the people with whom he has come into contact. And so I'd like to finish by paraphrasing yet another person who sent in a letter supporting this nomination, saying that Ed has made us all better scientists. So it's with great pleasure that I introduce this year's 2019 Fankuchen Award winner, Ed Lattman.
Thank you for the wonderfully generous introduction, and thank you all for coming to such an early presentation. Some of my early slides may be a little bit redundant with the introduction that you heard. At this stage of the proceedings, it's incumbent on the awardee to thank everybody involved, but in a relatively low-key way so that it doesn't seem too self-important. But actually, I'm thrilled. And so I really do want to thank everybody involved. It's particularly wonderful to receive an award from one's own professional society. The Nobel Prize in Literature is sometimes given for books that no one on the committee can read in their native language. And we all have a native language together. I'm also thrilled because the list of previous winners that you just saw, and that I'm going to show you again, includes many of my mentors and intellectual heroes in crystallography. And it's really an honor to be on the same list. So I'm going to talk a little bit about some of the interactions I've had with previous winners before I get onto the scientific content of my talk. So this is a picture that you already saw. You can see we weren't perfectly coordinated. Two pictures of Fan, as he was universally known. He had a wonderfully down-to-earth persona, which you might call a citizen physicist. And he was much more Brooklyn than Cambridge in his public interactions. And I remember vividly a seminar that Marvin Goldberger gave at Brooklyn Polytech about his ideas for determining phases by coincidence counting among the photon arrival times of three reflections touching a volt sphere at the same time. It was very arcane, and Van just demanded that he make us understand this seminar. Didn't actually work, but it was a magnificent effort. And I actually, in retrospect, understand that what Goldberger did is an outgrowth of the work of Hanbury, Brown, and Twist on radio astronomy interferometry, but I didn't get it at the time. In about 1960, Fan was persuaded to truck his equipment up to Woods Hole and to offer a crystallography session in the famous Woods Hole Marine Biological Laboratory Physiology course. And there were some bad moments, such as when the ocean invaded the basement where Fan's stuff was. The procession cameras didn't like it a bit. At the end of the course, there was probably still is, a wonderful lavish picnic. And at this particular version of the picnic, the students came up to Van with a special dish that they had created for him. And he regarded it with due suspicion. And he said, "What's that?" And I recreated it for you, and it is a reciprocal lattice and tomato sauce. So he took it well. So here's the list of winners that you just saw. As a very senior member of the community, senior is the euphemism that one commonly uses these days. I have had personal interactions with a vast majority of the people on this list, even going back to Martin Berger. And I'd like to talk about a few of them who were particularly important in my intellectual development one way or another. David Sayre. I had the rare privilege of becoming a close friend of David's later in life. Of course, I had been very much aware of his work, and indeed, I'd spent quite a bit of time as a graduate student re-deriving results that David had discovered a generation before. I should have read the literature. 1952 was quite a year for David. His paper, Some Implications on a Theorem Due to Shannon, which I'll show you in a moment, pointed out that if we could sample diffraction patterns at half-integral reciprocal lattice points, it would be possible to determine phases directly. And this is the ancestor of many contemporary methods, such as symmetry averaging and all kinds of other methods that let us oversample in ways that allow us to interpolate in the transform. Also in 1952, David published a critical paper on direct methods, deriving what's called Sayre's equation that's closely related to sigma 2 in the Karl Hauptmann formalism. And it's based on the physical insight that if you have point-like atoms and square the electron density function, properly normalized, the squared electron density function looks a lot like the electron density function. And this leads to a convolutional equation in diffraction space where you can solve structures. I always thought that David's work on this was not sufficiently recognized at a critical moment. So this is that paper. It has more citations than it has words, which is not that common. David left crystallography in the middle or late 50s and went to work for IBM for over 20 years, where he helped develop Fortran and did many other critical things that we're only dimly aware of because of the corporate environment. But he came back into crystallography, into imaging, I guess, to work on X-ray microscopy. And the title of his award lecture was on the possibility of determining the structure of a particle weighing 10 to the 12 daltons. Well, that foreshadows the efforts on single particle imaging with X-ray lasers today. So a prescient guy and a wonderful friend and mentor. This is the page, cover page from a book on solution scattering by André Guigné, a former winner, and his colleagues, which belonged to my thesis advisor, but which I liberated because he really didn't care. This was a very difficult book for me to read as a graduate student. And it's really hard to visualize how difficult these experiments were 50 years ago, 75 years ago, with weak tubes, crappy optics, and all the other things. And it's miraculous how much these guys got done. And this led to a book with my colleagues Tom Grant and Ed Snell, so that this is intellectual mentorship at a distance, but if I'd never seen Guigné's book, I'm sure I never would have gotten involved enough in solution scattering to do this. Finally, let me touch for a moment on Don Caspar, who was my postdoctoral advisor at Brandeis with Carolyn Cohn. So Don is the epitome of a deep thinker. He was the co-creator of the pseudo-symmetry theory of the surface lattice of icosahedral viruses. He did seminal work on TMV and various mutants, and he understood diffraction in ways that are very rare. He also included you in his intellectual life in a deep way. You would sit down with Don, and there was a topic you'd been thinking about for 30 years, and he would talk to you as though you had been thinking about it for 30 years too. And that didn't always work. When I got to Brandeis, I understood 10% of what Don said. And when I left, I understood 50% of what Don said, and I believe that was a record, which I'm quite proud of. Okay. So I'd like to switch now to the main topic of the talk, which takes advantage of the part of the award that says the recipient is alleged to be a good teacher of crystallography. And what I'd like to do is try and teach you a surprising amount about small angle X-ray scattering in a half an hour. And to motivate you, I'd like to spend a few minutes talking about a contemporary study using solution scattering that gains the kind of information that most people don't think that solution scattering can provide, and kind of insights. Another example of this was Tobin Zosnick's wonderful talk yesterday, and actually, in a well-ordered universe, this talk would have come before Tobin's, so that it would help those of you who haven't been playing with small angle scattering to dive into his talk more directly, but programs are random. Okay, so the small angle scattering, which is sometimes done at wide angles, but I'll call it small angle scattering anyway, is a technique that's very simple in principle. You have an X-ray beam that comes in, it illuminates a solution containing a particle. I'm going to call them particles even though they might be molecules. You have to call them something. And it's usually a dilute sample. It's 1% by weight specimen. So the signals are weak. Their molecules or particles are tumbling, and so you get a diffraction pattern that's circularly symmetric, and it's really a section through a three-dimensionally spherically symmetric diffraction pattern. And it can be completely summarized by the scattering curve, which is called I of Q versus Q. Q is the scattering vector used in solution scattering. It's 4 pi times sine theta over lambda in magnitude. Crystallographers often use S, and Q is 2 pi times S. So here's the study that I wanted to use to motivate what I'm going to try and teach you. This is work from a large group of people headed by Richard Neutze. It's on a photosynthetic reaction center from this organism, which I stumble over. And it has to do with the following observation. This is a light harvesting protein. It has antenna-like molecules that absorb photons. They propagate into the protein where they are made use of, but in addition, the protein is surrounded by lipid, and the lipid is surrounded by water, so it's a 4-component system. The amount of energy that you can deposit in these antenna-like molecules is far more than is needed to unfold the protein. So the question is, how does the protein get rid of all of this energy before it unfolds? And theoreticians have postulated a protein quake, which has a mechanism similar to how an earthquake dissipates energy. So here's their experiment. This is the interaction point. This is an infrared laser that will pump energy into the antenna molecules of the protein. And this is the X-ray beam, which is a pulsed beam from an X-ray laser, very short pulses. And you collect a solution scattering pattern at some time, known time, after you activate the molecule with light. And these are a series showing some of the curves that they collected. These are different spectra. Difference between the spectrum before illumination and after, 0 picoseconds, half a picosecond, and so on. The black curves are data, and the red curves are fits from a principal component analysis of the curves. I almost don't have to go on to convince you that a family of curves like this, evolving in time, contains a lot of information about what's going on with the protein. But I will go on a bit. They used principal component analysis. These, which are shown here, these are the Q curve, I of Q curves, for each of the 4 principal components. And these are their weights to build up the spectra as a function of time. They also carried out molecular dynamic simulations in which the molecule was heated to 3,000 degrees in the antenna portion, and then the heat was allowed to propagate through MD simulations. The structure of the unactivated protein was known from crystallography, but an EM structure would have been perfectly adequate for this. So this tells you what these 4 components are. I don't want to go through it in detail. C3, for example, represents non-equilibrated heat flow from the cofactors to the protein and lipid. So it's a pulse of heat going out, but it's not equilibrated because, for example, atoms that have temperature factors aren't all rapidly moving independently anymore. There are collective motions. This is a picture of 2 of the different components in which delta I is fit by the principal components. These two panels, which are actually a stereo pair, represent structures taken from a synthesis of many molecular dynamics simulations, which yielded scattering curves that agreed with the experiment. For example, the spheres represent atoms that move collectively together in most of the molecular dynamics trajectories that they ran. And what happens is those are atoms whose distances increase as the shock wave passes through the protein. So we learned a lot here. We validated the protein Quake model. I didn't talk about all of the different steps. The intensity curve calculated from molecular dynamics coordinates gives you typical coordinate sets for a pathway along this whole trajectory. There is no one trajectory for molecules, so you can't have a unique set of coordinates, but these are sample coordinates. A hidden nugget in here is that it appears that this work more or less validates the use of molecular dynamics potentials at very high temperatures, because they got answers that agreed with experiment, even at very short times. It's not an easy experiment to do, to check MD potentials at very short times. Okay, so there's an example of contemporary solution scattering experiment that could provide a lot of information that's of genuine interest, that's of high resolution, and so now how do we get there? So the first thing you need to do, if you think about those curves, is to know how to calculate structure factors. How do you calculate the diffraction pattern of a molecule that's rotating through all angles? So it turns out it's really easy. Instead of having a whole bunch of molecules, let's keep our eye on one molecule as it rotates in solution. If we could take snapshots of it every picosecond or something, we would have a set of snapshots that build up the whole three-dimensional diffraction pattern of one molecule. And all of these sections get averaged in the picture we take, so we realize that the diffraction pattern that we observe is the rotationally averaged diffraction pattern of a single molecule. The Fourier transform of the diffraction pattern of a single molecule, the intensity pattern, is the Patterson function. So the Fourier transform of the rotationally averaged diffraction pattern is the rotationally averaged Patterson function. So let's calculate the diffraction pattern starting from the Patterson function. Imagine we don't need a picture. You have one peak in the Patterson function. It's a position Rij, vector between scatterer I and scatterer j. The spherical average of this is a fuzzy spherical shell. If it were a point atom, it would be a ping pong ball. Because the atoms are fuzzy, it's a tennis ball. The Fourier transform of a spherical shell is a simple function. It's a sine x over x type function. It's one of the small pleasures of solution scattering. The Fourier transform of a sphere is not a Bessel function, it's a sine x over x. And so each peak in the Patterson function gives rise to a different spherical shell. And of course, they're incredibly heavily overlapped. But the diffraction pattern of one spherical shell is given by the atomic scattering factor of the two atoms times Q Rij divided by Q Rij. Total diffraction pattern is just a sum over all the interscattering vectors, all the Patterson vectors of the scattering factor times the sine x over x term for each atom. This is called the Debye equation. There are more efficient ways to do this calculation, but now we know how to do structure factors. So let's return to this curve. What I'd like to do next is take you through different segments of this curve. I'd like to start by a low angle part, low Q, in this vicinity. I'm going to tell you the answer first and then show you how we get it. So it turns out that the low angle part of the I of Q curve is very close to a Gaussian. And so if you plot I of Q, it is a Gaussian of this form where rg squared is the square of the radius of gyration of the particle. And I'll show you where that comes from in the derivation in the next couple of slides. But if you take the log of both sides, and you plot it against q squared, you get straight lines whose slope is equal to the radius of gyration squared divided by 3. Let me remind you that in solution scattering, the radius of gyration is measured by the mean square distance of the scatterers from the center of mass. In mechanics, it's often with respect to an axis, but in solution scattering, it's with respect to a point. So the radius of gyration of a ping pong ball is equal to the radius of the ping pong ball itself, because all the scatter is at the surface. The radius of gyration squared of a sphere is 3/5 of the radius of the sphere. So we're going to use our previous trick and try and derive this by going back to the Patterson function, see if we can't show that the Patterson function at low resolution is also Gaussian, because the Fourier transform of a Gaussian is a Gaussian. So if the Patterson is a Gaussian, so is I of q. So here's the thought experiment. This oval here is not the Patterson function, it's a structure, a simple structure. What we're going to do is we're going to try Patterson vectors of every length in a kind of random walk experiment. We take a vector, we put its end down at a random place in the object, we spin it at a random angle, we ask is the other end of the vector in the object. So this is where the rotational average comes in. So here's one where the vector goes out, here's another one where they stay in. And you do this over and over again mentally for every vector length. So if the vector is of 0 length, both ends are always inside. If the vector is longer than any dimension of the object, the other end is never inside. So this is a function that goes monotonically from 1 to 0. And because it's a random walk type problem, it's entirely plausible to write this thing as a Gaussian. Okay? And so if you do, the rotationally average Patterson function, p sub r, is proportional to this Gaussian, where r is the radius, sigma squared is the mean square distance between scattering centers. It would be atoms in an atomic model, but it could be balls if you have some kind of a low resolution model. This factor of 3 comes from the fact that 1/3 of the variance comes in along each direction. It also turns out that the mean square distance between scattering centers is closely related to the radius of gyration. There's a factor of 2 involved. I don't want to do the derivation. Let me just say that if you draw a vector from point I to point J, you can draw a vector from I to the origin, center of mass, and the center of mass back to J. And so you decompose the vector ij into 2 vectors that also go into the radius of gyration equation. So that's where, all right. So if we Fourier transform this Gaussian, we get that I of q is going to equal to I0 times this same factor. Rg is now in the numerator, and it gives us the Guignay plot that we saw before. So this is a way of showing that the low-resolution Patterson function is Gaussian, and then we Fourier transform it to get a low-resolution Gaussian for I of q, from which we find the radius of gyration. This summarizes where we are. I didn't mention, but it's not hard to show that if you extrapolate I of q as it goes to 0 to approximate I of 0, you can also find the molecular weight. Let me now talk about the part of the curve that's in this region over here. And for many particles, the curve past the Guignay region can be approximated by q to the minus n, where n has different values for different shapes. And I'd like to show you how you find n in a fairly simple and straightforward way. The resolution for this is defined as 1 over q is on the order of the diameter of the particle. So it turns out that at these resolutions, almost all the scattering comes from not within the particle, but in the contrast region between the particle and the solvent surrounding it. And this means that we can model I of Q just by modeling the surface of the particle, not having to model the volume. So let me show you how we can argue that the density, that the scattering occurs from these boundaries. So this is a picture of a specimen. The blue circle is a bit of the solvent, and the red circle is a particle. And the X-ray beam is coming in from here. Here's the scattering vector Q. I'm going to represent the density in this specimen as a sum of a density on a set of tiles, which are these little orange things. You know, we can break up the electron density in an object in any way we want and add it up to get the scattering. In crystals, we do it from atoms, or we do it from electron density voxels. And we're entitled to do it any way we want. So this tile has been carefully chosen by me to have two characteristics. One is that its long axis is parallel to Q. And second, that its length is pi over Q. So if we walk along one row like this, from this to this to this, there's a phase change when you go from one tile to the next. And we know that the phase change is Q dot delta X. And if we work it out, the phase change from this tile to this one is exactly pi. That's why I picked the length. And the phase change from here is to pi. So if you walk along each row, the scattering from each tile cancels the one after it, or before it, until you get to a place over here where this tile is in solvent and this one is going through the particle. And this one has a phase shift of pi, but it doesn't have the same density in it. So it's in this region that you actually get the net contribution to the scattering. Okay, so here's two scattering curves. The one in red, the one in blue is from a sphere, solid sphere. The one in red is from a spherical shell. So I argued that all the scattering comes from the surface, but these things don't agree very well. However, if we instead use a shell at the radius of gyration and not at the particle surface, you get very close agreement between the scattering from the solid object and the scattering from the surface representation. So at this kind of resolution, it really is fair, if you consider the shell to be at the radius of gyration, to work with the surface. This is, I think, a new derivation. I actually haven't seen this in any book. It's special for you. So now, let me give you an example of how this works with a sphere. It turns out that the sphere has intensity that falls off as Q to the fourth spherical type particles. So the big gray circle is a sphere that we're looking at in cross-section. And we're covering its surface with little balls, atoms or scatterers. And the idea is to calculate the scattering from this set of, I'll call them atoms, even though they're much bigger than atoms. And the average intensity, as it is in crystallography, is equal to the number of atoms times the square of the atomic scattering factor. The square of the atomic scattering factor at low resolution is just the square of the number of electrons in the ball, which is proportional to the square of the volume of the little ball, the atom. So how many atoms are there? You can figure out how many atoms there are by taking the area of the whole particle and dividing it by the area, cross-sectional area of one of the little balls. So the area of the big one is proportional to r squared. The area of the little one is proportional to 1 over q squared. And so the number of atoms is proportional to q squared times r squared. The atomic scattering factor is proportional to the volume of the atom, therefore, because that's filled with electrons. And so that's proportional to q to the minus 6th. And so I of q is proportional to q squared times q to the minus 6th goes as q to the minus 4th. You can do the same argument for a rod, and you get q equals q to the minus 1. The disk gives q to the minus 2. So Otto Kratky created a plot to help distinguish these families of things. It plots q squared times I of q, so that a q to the minus 1 curve will continue to rise, a q to the minus 2 curve will plateau, and a q to the minus 4 curve will still fall. And this is an example of a Kratky plot taken from our book, and this is q squared times I of q. And I don't want to go through all of these curves because there was a long introduction, and I don't want to run over. But here's an example of a q to the minus 4th. Here's an example of q to the minus 1. Unfolded proteins can be approximated by a set of flexibly connected rods of the persistence length of the protein. And these other ones represent samples that have more than one domain that can behave differently. Okay, so this is what we learned. We know the structure factor equation. We know that at low resolution, I of q is Gaussian, and that we can get the radius of gyration. At medium resolution, scattering occurs mainly by contrast between the particle and the surface, and you scale by RG for the best agreement. This lets you get the q to the minus n dependence for a variety of shapes. So that's quite a bit from solution scattering, but I wanted to finish with one topic from crystallography, just because it's the ACA after all. So this has to do with the temperature factor. So in this diagram, if I can hold my laser pointer still enough, the vector over here represents the contribution to the scattering at reciprocal lattice point H from one chosen atom in a chosen unit cell, traditionally named J, right? Our old friend J. So the amplitude of that scattering is the atomic scattering factor Fj, and the phase is given by 2 pi h dot xj. The other vectors in this diagram represent scattering from corresponding atoms in nearby unit cells. By corresponding, I mean atoms related by a lattice translation. And as a consequence of the von Laue interference condition, or maybe even an alternate statement of it, that these atoms are all in phase with the one we chose at our origin. And so you get constructive interference of lots of these vectors and strong scattering. And actual crystals, of course, our atoms are in motion. And that's discussed here. I've left the atom in the first unit cell undisturbed just for reference. But atom 1 has undergone a shift delta x1 and so on. And these phase shifts, these position shifts, turn into phase shifts. So the arrows from these things rotate back and forth as delta x changes. And so instead of these things being fully coherent, they make a kind of tipsy walk around the correct position, or the mean position. And qualitatively, this looks just like a temperature factor. As h gets bigger, the wobble gets bigger, and the projection onto the mean length gets smaller. And again, as delta x gets bigger, a bigger b factor, again, these things wobble more and more and more, and they give you a smaller and smaller projection in this direction. But it turns out that you can also get the quantitatively correct answer. The factor that relates the tipsy walk intensity, amplitude, to the normal amplitude is mean value of cosine of 2 pi h delta x. And if we assume that delta x is Gaussian, we can compute the expectation value of that by saying, here's cosine 2 pi delta x, here's a Gaussian distribution for delta x. And I don't want to go through the rest because I'm falling behind. But again, the Fourier transform is a Gaussian is another Gaussian. And this one is our old favorite, e to the minus b sine squared theta over lambda squared. Won't go through the algebra to translate from h to that. So the temperature factor comes about because of this tipsy walk of the vectors that are adding up. Let me conclude with one last thing. Michael Rossman was a wonderful mentor to me. I spent a week in his laboratory once when I was a very junior graduate student, learning to use his rotation function program. He was gracious throughout my life. He was a wonderful advisor, and of course, an acute critic when the need arose. Sometimes in people's careers, one gets an opportunity to write a nomination for a Nobel Prize, and I wrote one for Michael, and I'd like to read a little section of it in my concluding slide. As one of the founding fathers of macromolecular crystallography, Michael Rossman has made major and continuing contributions, both to the theoretical and experimental aspects of the field and to the structural biology of the systems his laboratory has studied. His nomination could have been based on a number of contributions, but the human rhinovirus system was chosen because it combines technological tour de force with the extension of basic chemical principles to a whole new domain, the pathogenesis of viral disease. Finally, I have some acknowledgements that you can read, and I thank you for your attention.